Next slide, please. So I will start today's presentation by a quick overview of NBTXR3, or what we call R3, a first-in-class radioenhancer. R3 is made of hafnium oxide nanoparticles, and thanks to high electronic density, hafnium is an excellent energy absorber. R3 is inert without ionizing radiation, but activated when irradiated. Its negative surface charge and its nanometer scale facilitate entry of R3 into tumor cells. When injected into a tumor, which is done one time before starting radiotherapy, R3 allows for a higher level of energy from radiotherapy to be absorbed and transferred to the tumor. R3 has been shown to be safe and effective in soft tissue sarcoma, and we believe that its mechanism of action is universal and active in all tumor types tested. Next slide. As mentioned, R3 is able to increase energy absorption in any solid tumor after injection. This has two effects. First, it amplifies cancer cell destruction in the RT-injected tumor, improving its local control. But data suggests that R3 is also able to trigger a systemic effect, which seems to affect no-injected tumors outside of the field of irradiation. We believe this effect is mediated by the patient's immune system. Next slide. Next slide, please. Yeah, thank you. As you can see here, we are and have tested R3 in several solid tumor types. We have successfully demonstrated the benefit of R3 activated by radiotherapy compared to radiotherapy alone in patients with locally advanced soft tissue sarcoma. So far, hundreds of patients were treated, showing safety, feasibility of this approach, and consistent tumor response. In addition, R3 not only appears to be tumor agnostic, but also can be combined with other therapies like checkpoint inhibitors or chemotherapy. In fact, we believe that any patient who is going to receive radiotherapy is likely a candidate to have that radiotherapy enhanced by R3. Next slide, please. As you can see from our clinical trial pipeline, there is still a lot of work to do. We are working closely with J&J to develop trials primarily focused on head and neck and lung cancers. However, you can see at the bottom part of this slide, there are also many opportunities for expansion into other indications. Next slide, please. So, as I said before, there are two mechanisms by which R3 acts: direct cell killing and priming the immune system. And I'm going to describe this effect in detail now. Next slide, please. So this slide summarizes all the different steps followed by R3 from intratumoral injection and internalization by the cell through radioenhancement to cell destruction. Thanks to its unique mode of action, R3 can induce multiple biological responses. Next slide, please. Among the steps, certain biological responses like lysosomal membrane permeabilization, observed only with RT plus R3 in our models, as well as amplified DNA damage, and certainly other biological effects, ultimately lead to increased cell death and can be associated with the physical and pleiotropic mechanisms of action of R3. Next slide, please. What is also very interesting with R3 is that beyond its capacity to amplify cell death, the biological responses produced can also participate in activating the anti-tumor immune response. This is the case, for example, of the improved formation of micronuclei, inducing the production of interferon beta, known to be a potent activator of the immune system. This is also the case for the increase in the amount of peptides presented on the cell surface by MHC1 complex, the increase of the immunogenic cell death, and a greater release of antigens, facilitating the activation of the adaptive immune response. Next slide. To illustrate the direct cell death enhancements of R3, you can see on the left some of the efficacy experiments performed on 15 different cancer cell lines, showing that the addition of R3 activated by radiotherapy amplifies cell death, compared to radiotherapy alone. This enhancement of cell death results in a better local control of the tumor in vivo, as can be seen on the right in some of the 11 models tested. Next slide, please. On this slide, you can now see some of the preclinical results illustrating impact of R3 on different biological pathways, such as DNA damage and induction of the Sebastien pathway, leading to secretion of interferon beta, the immunogenic cell death, with here represented the relocalization of calreticulin to cell surface membrane, or the modulation of the immunopeptidome required by CD8 T cells to identify and specifically destroy tumor cells. All these pathways participate in the priming of the immune response. Next slide, please. The priming of the immune response by R3 is illustrated by this study. Immunocompetent mice bear a tumor on each flank. Only one tumor received a treatment, the other remained untreated. On the left, we can see that at the level of the treated tumor, R3 plus radiotherapy induces a slight benefit compared to radiotherapy alone. But if you look at the untreated tumors, only the mice in the R3 plus RT group saw the growth of their tumor significantly slow down, while RT had no effect. This better local control of tumors, of course, significantly increases the survival of mice. As we can see on the right part, this effect is linked to better CD8 infiltration in both tumors, only in mice from the RT plus R3 group. We can see here that only the mode of action of R3 and the quality of cell death are able of inducing an immune response strong enough to produce a systemic effect, while RT alone cannot. Next slide, please. This ability of RT-activated R3 to produce anti-tumor immune response made possible to overcome anti-PD-1 resistance. In the study presented here, cell resistance to anti-PD-1 were implanted on each flank of immunocompetent mice. As previously, only one tumor received treatment with RT on R3. The other did not. Mice also received anti-PD-1 treatment. You can see at the bottom in the green curve that the combination of RT plus R3 and anti-PD-1 provides better control of both tumors compared to RT plus anti-PD-1 in blue. This better local control of tumors significantly increases the survival of mice and drastically reduces the formation of lung metastasis. Next slide, please. Other combinations of checkpoint inhibitors have given similar results. Strong priming of the adaptive anti-tumor immune response, overcoming resistance and offering both local and systemic control, and decrease of spontaneous lung metastasis. Next slide, please. Another very interesting point regarding the combination of R3 plus RT with CPI, with checkpoint inhibitors, is the ability to produce a long-lasting memory response, similar to what can be obtained after vaccination. In this study, mice in the R3 plus RT and a combination of CPI group saw both tumors completely disappear. After several weeks without signs of relapse, these mice were rechallenged with a new injection of tumor cells. No mice developed a tumor, unlike the naive control group, where all mice developed a tumor. Next slide, please. And finally, here we show the translation of what I just detailed before for this particular patient. With a single injection of R3 before the patient's course of radiotherapy, they achieve both local control at the site of injection and irradiation, but also a full complete response at the distal site of disease that did not receive R3 nor radiotherapy. And these results were seen in patient who is anti-PD-1 resistant. So you will hear more about these kinds of results in the next two presentations. And with that, let me turn the call over to Dr. Harry Rosenberg.